I thought this was really funny. You hungry? I can use a light snack. Okay, so what is photosynthesis? This is the process in which light energy is transformed into chemical energy. And so this is a really big part of the study design, so you do need to know it in quite a bit of detail. Here you've got thylakoids. So thylakoids are where the light-dependent stage of photosynthesis occur. And then around it you've got stroma. So stroma is the fluids-filled space, and this is where the light-independent stage occurs. So you've got two stages of photosynthesis, the light-dependent and light-independent stages. And they're kind of separated depending on where they actually occur. So what is a chloroplast? A chloroplast is an organelle, which is the site of photosynthesis. It's got two membranes, so it's a double membrane bound organelle. And within the chloroplast, you've got grana. Grana are stacks of thylakoid discs. And so you've got the site of light-dependent stage occurring here. You've also got stroma. So stroma is the fluid-filled space around the thylakoid stacks. It's a site of light-independent stage. So you can see the chloroplast here. You've got two membranes. You've got the stack of thylakoids. You've also got stroma and So thylakoids within their membranes contain photosynthetic pigments known as chlorophyll, and these are the photosynthetic pigments which absorb light in photosynthesis. The various types of chlorophyll, such as chlorophyll A and B, you don't need to worry too much about them, but these different types of chlorophyll absorb different wavelengths of light. I don't think you need to know too much about this here. You can see that you've got a high amount of light absorption occurring at like blue light and a high amount at like red light, but not a lot of green light. This is because the plant actually reflects the green light and doesn't actually absorb it, and that's why they appear green, because they've reflected the light. Okay, so there's a really important enzyme involved in the in photosynthesis in the carbon cycle, which is a stage of photosynthesis. So its role is to fixate the carbon molecule from carbon dioxide into an organic molecule that the plant uses for energy storage. So during the carbon cycle, Rubisco fixates carbon to become the organic molecule G3P, which is a 3-carbon molecule, or half of a glucose molecule. So 2 G3P altogether combine to make a singular glucose. And during this fixation process, carbon dioxide will actually react with this 5-carbon molecule called RuBP to make a 6-carbon molecule that splits into 2 G3Ps. Kind of a complex process. Once again, this document is a lifesaver. I haven't actually taught photosynthesis this year yet, but every year it's kind of like it's just so intense. Let me just control for Rubisco. Oops, Rubisco enzyme. There it is. So this kind of summarizes what we need to do about Rubisco. So we need to know that it's got a key role in the carbon cycle in photosynthesis, and we need to be able to explain how plants have adapted to maximize this efficiency of photosynthesis. So in both carbon dioxide and oxygen can bind to Rubisco's active site, but at low temperatures, carbon dioxide is more likely to bind to oxygen than bind to Rubisco. So really understanding that Rubisco is kind of a temperamental enzyme. I actually remember looking it up last year with my students, and the website described it as kind of being kind of a poor enzyme. Like, it doesn't do that well. Like, what it's supposed to do is it's supposed to bind carbon dioxide, but it can actually bind oxygen instead. So then this is called photorespiration. So photorespiration is not useful. We don't want photorespiration to occur, but Rubisco is kind of just a poor quality enzyme that apparently can bind oxygen instead, and this is not useful. So yeah, definitely going back to this document here and trying to revise it, understand what's going on with Rubisco. Yeah, that's really important. I think I've just kind of summarized quite a bit here. So you can see here, you don't need to know too much about all of these stages, but do understand that you've got carbon fixation occurring, and you've got the production of 2 G3P molecules, also known as GA3P. And so GA3P or G3P produces half a glucose molecule. Putting two together creates one whole glucose. Okay? And you've also got some ATP being consumed in this reaction. I don't think we have enough time to really explore this in any more detail, just knowing that there's a light independent reaction, also known as the Calvin cycle, and yeah, you've got Rubisco enzyme having a key role making half a glucose molecule, known as a G3P, GA3P. This kind of just summarizes the inputs and outputs of photosynthesis. So water, ADP, and inorganic phosphate, as well as NADP, are inputs, and an output of light-dependent stage are oxygen, NADPH, ATP. This occurs in the grana. The second stage is the light-independent stage, and you've got carbon dioxide, ATP, and NADPH as inputs. Outputs are glucose, ADP and PI, NADP+, and this occurs in the stroma. And then you just get a summary of the formula once again, and make sure you do know this formula completely. Like memorize it, which I know is annoying to have to like memorize stuff, but it is very important to make sure you're on top of it. Okay, some more water. So most plants will actually undergo photosynthesis via the pathway that we just looked at. These are C3 plants. So there are different types of plants. Under typical conditions, photosynthesis works pretty well. However, in C3 plants, it can sometimes be inefficient. So as I mentioned before, rubisco can actually undergo something called photorespiration. And so instead of fixing carbon, as it should, it might accidentally fix oxygen instead. And so this is kind of wasteful. It leads to photorespiration, and it wastes energy and uses up carbon molecules, making photosynthesis really quite inefficient. So as I mentioned before, rubisco is kind of like not an amazing enzyme. Like it gets the job done, but like there's lots of room for improvement. So photorespiration occurs when carbon dioxide levels are quite low. Okay, so you're a plant and you're in a low carbon dioxide environment, you might actually undergo photorespiration and start fixating oxygen instead of carbon dioxide, and this will waste your energy. We've got a few different types of problems, I guess, that we need to overcome. So different types of plants, so not C3 plants, C4 plants rather, such as corn. These are usually found in hotter areas, and in order to minimize water loss, C4 plants often have their stomata closed. And so stomata are like little holes, pores on the side of the plant. If it's in a really hot area, plants such as corn will close their stomata. While this is good at preventing water loss, it can actually be quite detrimental to the plant because it can lower the amount of carbon dioxide entering the plant. As such, you may actually have photorespiration occurring, which is that fixation of oxygen. Not good. So we have to get around this somehow. And so plants have actually kind of evolved to get around this. And so what C4 plants do is that they separate the light-dependent and independent stages into different cells within the plant. So you've got two types of cells, mesophyll cells and bundle sheath cells, which are kind of involved in this process. And so in a mesophyll cell, carbon dioxide is fixed into a molecule called malate. Okay? So carbon dioxide is fixed into a molecule known as malate. This malate will then be transported to a bundle sheath cell where it will release carbon dioxide, and that will then be fixated by rubisco. So just kind of to emphasize this, if you're in a hot area, you might actually lose water, so you shut your stomata, but this can prevent carbon dioxide from getting in, right? So you might actually have photorespiration likely to occur. And so in order to get around this, you can separate your two stages of photosynthesis, light independent and light dependent stages. So, in a mesophyll cell, carbon dioxide is fixed into a molecule known as malate, and then that malate will be transported to another cell, a secondary cell called a bundle sheath cell, where it will release carbon dioxide, which is fixated by rubisco. This means that even when it's hot, C4 plants can have their stomata, close their stomata, and still have a supply of carbon dioxide for rubisco, preventing photorespiration from occurring. So you can see here the separation of photosynthesis into its two stages into two separate cells can be quite a useful process to kind of circumvent that loss of carbon dioxide that you get from shutting your stomata. Can plants function in a similar way to C4 plants and are adapted mainly to dry environments. Instead of separating locations, can plants separate the light independent and dependent locations or reactions over time. Not locations, sorry. So in C4 plants, corn and other starchy plants, they separate those two stages via actual physical locations. With can plants though, you actually separate them over time. And so you open your stomata at night when it's cooler and more humid, and this allows the carbon dioxide to actually enter the plant. That carbon dioxide will then be converted into malate, which is then stored in the plant until the daytime. During the daytime, your stomata might close and you might not be getting enough carbon dioxide, but that's okay because you've already got the malate there, and that malate will then release that carbon dioxide, and this allows rubisco to fixate it during the carbon cycle. Okay? So keep in mind, can plants have separation of the light-dependent and independent stages of photosynthesis temporarily temporally. I think it's the right word, temporally, or over time, whereas the other one is like a location-based differentiation. So cam plants can actually close their stomata during the day. It still have a supply of carbon dioxide for rubisco, preventing photorespiration from occurring. So they open their stomata up at night, let the carbon dioxide in, store that carbon dioxide as like malate, and then that malate will be open up later on during the day once the stomata is closed. So you see the stomata is closed during the day, but you still have sufficient amounts of malate, allowing photosynthesis to occur as opposed to photorespiration. Kind of a lot of content. Use a drink. Okay. Cool. If you've got any questions, just like pop them in the chat. I know it's kind of an intense area of study. It's kind of a lot going on. So let's put them in the chat and I'll get back to you as soon as I can.